Okay, so our first presenter will be Dr. Katie Birnie, and she is a clinical psychologist and assistant professor in the departments of anesthesiology, perioperative, did I say that right? Perioperative and pain medicine and community health sciences at the University of Calgary. She leads the Partnering for Pain Research program that uses patient-oriented research methods to improve the prevention, assessment, and management of pain in children and their families. Our second presenter today will be Sarah um, Merrill. Sarah Merrill is a neuropsychologist who is using social epigenetics to understand the effects of early environment on biology. She received her PhD from Cornell University in developmental psychology and is currently a postdoctoral fellow with Dr. Michael Kobor at the University of British Columbia. And then lastly, we have our fabulous Dr. Kharah Ross, who is a health psychologist with a specialization in maternal child health and psychoneuroimmunology. That's basically the study of connections between psychosocial states such as stress, close relationships, and the immune activity. She is an assistant professor in psychology at Athabasca University and an adjunct faculty at the University of Calgary. So we'll start with Katie. So please go ahead, Katie, and I will stop sharing my screen.
Wonderful. Thanks so much for that introduction, Martha, and really pleased to be here with you today, and grateful to Kara for the opportunity to actually talk a little bit about how we can make the blood sample collections as part of these studies in pediatrics that look at epigenetics and other factors easier. I remember sitting with Kara in the cafeteria at Alberta Children's having a conversation about this topic. What seems like eons ago now, but is a topic I've actually had conversation with a number of different child health researchers since that time, and really see a lot of value for how we can make research methods as comfortable and distress-free for children. So I just want to start by saying the painful needle procedures in particular, if that's how blood is being collected, but all types of different needle procedures, they're very common in medical care. They're common in the context of blood collection for youth, and they actually can cause a lot of distress. And even for children who undergo these quite frequently, this can be the greatest source of distress in medical care, even for children, for example, with cancer, who maybe undergo a lot of different types of medical interventions. And the onset of needle fears can start in early childhood and continue over the lifespan and lead to significant avoidance of medical care, are described as traumatic events, and so on. And so in the context of research studies where we're doing this, we really need to be making sure we're minimizing the distress that's connected to these experiences. The other part I want to add is that when we're working with kids in research or otherwise, it's not just the kids that are experiencing or can be impacted by or bring their distress to the experience, also their parents. So you can see this is a picture of a baby receiving an immunization number of years ago, and the child actually looks pretty calm. He's looking at the health professional. It's actually the dad who's experiencing a lot of distress in this moment. So we need to not just consider the child and the child's behavior and coping strategies for them or prevention strategies for them, but also for how we deal with parents. The other part I just want to say is I think pain as a potentially adverse event or as a neglected adverse event, or the real importance of managing pain around even basic kind of commonplace procedures is really increasing in attention. So this article from CMAJ was actually published in 2010, but talked about pain as a neglected adverse event, and medically caused pain, or in this context, potentially research caused pain, such as procedural pain. It's kind of our dirty little secret that we pretend is not an adverse event, but can be. And you know, when something kind of hits the New York Times, that it's being talked about, and it is really being identified as a critical issue. And this article is actually from 2018, maybe early 2019, as part of a series on pain in children, talking about how we actually need to make sure we're preventing and reducing pain from common procedures. I work as Assistant Scientific Director also of a national knowledge mobilization network. It's funded by the Networks of Centers of Excellence, called Solutions for Kids in Pain, or SKIP. And it's based at Dalhousie University, co-led by Children's Healthcare Canada. And we're really focused on mobilizing evidence-based solutions for children's pain through coordination and collaboration. And I'll just give a brief introduction, but why I want to share this is that actually we have an abundance of research evidence about how to do this well, how we prevent pain in children from procedures, as well as other types of pain. But often the gap is that people don't know about them, or they're not actually being put into practice.
Canada is a world leader in children's pain research. But did you know it can take up to 17 years for research findings to be put into practice? That's a whole generation of children who miss out on the benefits of information we already know. What we know now is that 2 out of 3 children in hospital undergo painful procedures without any pain management at all. 75% of parents say they don't know how to manage their children's pain. Veterinarians get 5 times more training in pain than people doctors. And only a few Canadian health institutions have made a certified commitment to better pain management. This matters because pain that isn't treated well harms children's brains and bodies, is a struggle, and can be traumatic for families and health professionals, and costs the healthcare system time and money. Scientists have found many ways to manage pain, but most people don't know about these treatments. That's why at Solutions for Kids in Pain, or SKIP, we are working to improve children's pain management across Canada. We are a national knowledge mobilization network that is bringing together researchers, patients and caregivers, health professionals, 48 different health institutions, including all the children's hospitals in Canada, and over 100 partners from around the world. Our goal is to get knowledge about children's pain into the hands of people who need it faster. If you're a scientist or researcher, patient, caregiver, health professional, policymaker, or part of an organization or company with unique expertise or technology, you can help. In fact, anyone can. We all have the opportunity to be a champion for kids in pain. Visit kidsinpain.ca to find out how you can get involved.
So really when we're looking at, so this is a great audience to be talking to, because this is another place in the context of research or child health studies where we can actually be implementing some of these preventative pain strategies around blood sample collections. And really our management can look at 4 different types of strategies. So pharmacological, so any medications or anesthetics, as I'll talk about, physical strategies that a child and a parent can be using, procedural strategies, and psychological strategies. And it's really the coming together of these that really helps to prevent pain in the context of procedures. So as the SKIP video suggests, there is an abundance of research evidence to support these different pain management strategies for procedural pain, such as needle procedures and otherwise. I've just put up, they've contributed to clinical practice guidelines for injections, but also there are a number, many Cochrane reviews that are synthesizing hundreds of randomized control trials and otherwise that really support these evidence-based strategies that I'll share with you. And you'll start to see many of these are actually pretty simple and don't require a lot of funding or aren't complex to implement, so can really feasibly be integrated into a research protocol. And strategies really can be broken down into what can families or children do ahead of time, what can they do at the time of the appointment, and what can they do during the procedure or during the blood sample collection itself. So from a procedural standpoint, if you're doing multiple kind of procedures, it's best to do the most painful last. Some of the physical strategies depend a little bit based on age, but for infants and younger children, breastfeeding both before, during, and after the procedure can reduce pain quite a bit. And this kind of is important to distill a myth that if mothers are breastfeeding their infants before or during a painful procedure, that somehow the infant might bite or, choke or something like that. Those are myths. There's no evidence to suggest that happens. And actually that breastfeeding, both the skin to skin and comfort positioning that comes with that, but also from the breast milk itself, can make a difference and can also more quickly help them recover in terms of any distress after the procedure is over. In the absence of breastfeeding, something called sucrose or other sweet tasting solutions can be placed in the mouth right before the procedure itself and can help to reduce that. And another option is sucking or a pacifier for babies or young children before, during, and after the procedure. Comfort positioning, even for older children, can make a difference. So for infants, that skin to skin against a mother's chest or a father's chest even can make a difference. Making sure that children are sitting upright before, during, and after. And you can also do comfort positionings like having children in a hug, facing their parents or facing away, but sitting on a parent's lap in a hug that can make quite a big difference. We really have to move away from children lying down. That increases that loss of sense of control and can increase fear. And under no circumstances should we be holding down children during a painful procedure or potentially painful procedure. There are devices. One's called the Buzzy. It's a vibrating device that goes just above the location where the procedure will happen, such as a needle or blood draw. And it also it vibrates and it also has some cold to it. So it's thought to prevent pain or interfere with pain in a couple of different ways, both as a distractor, but also interfering with some of the physiological kind of nerve signals because of the vibration and temperature. And then there are particular strategies that can be used for children about 7 years and older if they have a history of fainting. And I'll describe that one in a bit more detail. But essentially, it's a brief tense and release of muscles throughout their body. And that can come because, especially if the needle is involved, some people have a vasovagal response that can cause them to faint. In terms of psychological strategies, again, these are quite simple. A distraction, which essentially is thought to occupy your brain, giving less space to perceive pain, can be quite helpful. These can be any number of things: listening to music, watching a video, such as like on a parent's smartphone, or playing a game, even just talking with the child about something fun, like planning their birthday party or something like that. Or using deep breathing with a toy, such as blowing bubbles or a pinwheel that kind of spins, can help to distract them and relax the body with decreased pain. And another important thing is just the language that we use. So often our instinct is to reassure the child, say, this won't hurt at all, you'll be fine, I'm sorry this is happening to you. But what we've learned is that can kind of cue the child that there is something to be worried about, and actually have the reverse. Instead of decreasing pain, it actually increases anxiety and pain, kind of signaling that there is something to worry about. And so we suggest using more neutral words, and also not pretending like it may not hurt. Better to say, everyone experiences this differently. Let's see how you can tell me how it is for you. Or something kind of like, some people say it feels like a small pinch, but it lasts very briefly and then it's over. Better to say that than this won't hurt at all, because that can lead to a lot of distrust because it may or may not be true for that child. Pharmacological strategies, there's lots of evidence for topical anesthetics, such as EMLA and Amatop. They need to be placed in the area where the procedure will happen about 20 to 60 minutes before, depending on the product. Can be purchased over the counter, but in the context of a research study, maybe something you just have a small budget around or reimbursement for to families. And again, this is one of those myths. There's no evidence. There's a lot, I hear a lot about, kind of makes it harder to draw blood when topical anesthetics are used, but there's not really any good evidence to suggest that it constricts the veins or causes any issues. So certainly this is a strategy some families can be really effective. And of course, education. So the more we empower researchers, research staff, as well as parents and caregivers themselves and children about these strategies, starting that much ahead of time can make a big difference. And then for children 0 to 10, really having the parent present, engaging in some of that comfort positioning, engaging them in how they can distract the child and so on, can make a really big difference. So again, not overly complex or expensive things to do. but things that you can absolutely build in into a research protocol very easily to help empower families and make this an easy thing. Critically valuable if you're asking them to come for repeated blood collections over the course of a longitudinal study design. If you're interested in reading more, there's Denise Harrison is a pediatric pain researcher who used to be in Ottawa. She's now back at home for her in Australia, but she has a knowledge mobilization program called Be Sweet to Babies, which is really focused on reducing infant pain during painful procedures, and some great videos online available in lots of different languages. Dr. Christine Chambers initiated her It Doesn't Have to Hurt campaign in collaboration with a number of different organizations, including YMC, which was really geared towards creating information for parents that they can use. She uses the It Doesn't Have to Hurt hashtag, so feel free to check that out. We also use that now with SKIP. Christine is the scientific director for SKIP. Great videos, as well as got lots of great social media content geared towards parents. Here at Alberta Children's, they've synthesized all of these strategies in something called Commitment to Comfort. And actually, if any of you are getting your COVID vaccines right now, this strategy's been gone province-wide and gone across the lifespan. So it's available and part of our COVID vaccine rollout policies done by some incredible leadership of pain champions here at Alberta Children's. And they also have something called a comfort care plan. And so this, you can see was actually filled out by my daughter. She's 7, with her strategies that she wanted to use in preparation. She gets a bit nervous before needles. We've had some bad experiences in the past, despite being a psychologist who works in pediatric pain. So it really allows them to be planful, to be engaged, to create a coping plan before a procedure, and think about all the strategies connected to that. There's lots of resources online. We've worked with Parents Canada to do inserts for families. The Meg Foundation has some great handouts around different types of comfort positions family can use. And I've put a link, we've put together some of those resources on managing needle pain and needle fears on our website as well, that links out to a number of different pieces available for families. And I just want to show you some of the other pieces we put together. It's really simple to communicate some of these strategies to families that hopefully with some of these resources could be built into a research protocol.
As babies and toddlers, I always breastfed my kids during needles.
When my daughters were babies getting their first vaccinations, I would breastfeed them.
My younger son is often sick and needed hospitalization. I usually talk to him about what's going to happen and why we need to do it.
Most of the time, they hardly noticed, or they were only upset for a few seconds.
Using the numbing cream prior to the needle works wonders for this little guy. He just thinks he's hanging out with the nurse at the clinic and keeping mama company.
The cream or freezing spray helps me a lot, and sometimes whenever it doesn't work, I usually have the iPad with me.
Now that they're older, I give them videos on my phone to look at, or I coach them to look away, take deep breaths.
It really works.
I'm not scared of needles anymore.
We show her breathing techniques. We distract her while she's getting a needle. What do you usually like to use?
The phone.
The phone,
And
yes.
Gotcha.
Yes. And of course, numbing cream has been our saving grace.
My son's a little bit afraid of needles. So we talk about things that are interesting to him, and we always plan a special ice cream date for after. So we talk about what flavor he's gonna get.
Like cookies and cream.
Like cookies and cream?
So also building in rewards and other things that kids can get after the fact can sometimes help them a little bit more easily as well. And I just wanted to make a quick comment about needle fears, if that's how you're collecting blood. Needle fears are very common, so they impact more than 60% of children, as well as up to 50% of adolescents. And even if you're collecting blood with a needle venipuncture in children, it's likely that also 20 to 30% of parents of adults also have needle fears. But this real significant needle phobia is about 5 to 10% of the population. But we do know that poorly managed pain, poorly managed distress can lead to the development of high needle fears, particularly the typical onset is kind of that 4 to 6 year old age range. and that needle fears do contribute to avoidance of medical care and actually also vaccine hesitancy, which is a bit outside our scope today, but important to know. There are specific strategies for needle fears. The strategies I talked about before are more in preventing pain, but for those who've developed a real phobia, there are different strategies that are needed. And in the context of a study where there may not be direct benefit to the child, you really need to consider, how do we assess for the presence of needle fears, and how do we know that this actually isn't something we need to do or should be doing with this child, as it might contribute to really avoidance of their medical care more generally. But also, if they do want to participate, what could we direct them towards so that they can actually have some strategies to really address significant needle fears that's causing a lot of distress, avoidance, and so on. Yeah, so some resources for parents, articles for parents specifically about that, and also knowing parents are going to have some of these needle fears, and so their distress can, if they're worried about a blood draw or blood collection for their child, it's likely that their anxiety is going to be communicated and shared verbally or non-verbally to the child as well, and make the whole situation more distressing. SKIP that I do work with, we do have a program called Jump In with SKIP, where we do knowledge mobilization consultation with researchers and partnership. I think this is an area, even for people who don't identify as doing pain research in children, if there's ways that SKIP or myself can partner with you to develop research protocols that build in some of these simple pain management strategies for children as part of blood sample collection, I think, like I said, can be easy, relatively inexpensive, and help it go smoother for everybody. So just thanks very much. And please feel free to reach out. I'm on Twitter, but also by email.
Excellent. Thank you so much, Katie. That was a wonderful presentation, and especially so relevant with all of us getting our COVID-19 vaccination. So, and now we are going to hear from our, okay, so just a minute here. Kharah, are you going to be our second speaker today? Okay, so it will be Dr. Kharah Ross, and then we'll hear from Sarah Merrill after. Go ahead.
All right, so thanks, Katie, for getting us started here. So where I'm going from this is, now we've talked a little bit about how we get the samples. I'm sure that there's a number of people on the call who are kind of thinking, well, why do we need blood samples? Why is this a part of an intervention that's looking at child developmental outcomes? So for my portion of the talk, I'm going to give a bit of a higher order introduction to the theory, why we want these blood samples, what the information can tell us, and zoom in on a specific biomarker called RNA. So as a health psychologist, I am very interested and very aware of this link that has been very well established now, that exposure to chronic or toxic stress does put people at risk for poor health outcomes across the board, whether that's early mortality, development of diseases, things like that. And in the context of children, exposure to chronic or toxic stress can also potentially affect their health trajectories across the lifespan. So we know that this is something that's been going on. How is it happening? What is going on? How is this stress getting under our skin to affect our risk for health and disease later on? So there's a number of different pathways or mechanisms by which this can work. There are a number of indirect pathways. So for example, if you're exposed to chronic or toxic stress, you're probably engaging in not so healthy behaviors. So children in some of these environments, they might be more likely to be eating less nutritious food, which can have an impact. Children could potentially be more sedentary, especially if they live in a neighborhood where it's not safe to go outside and play, or they have a parent who's holding down multiple jobs. There just might not be the resources for them to go and play in these intramural activities. Older children, you have to worry about other health behaviors, for example, smoking, that could potentially affect their risk for disease later on. But there's also a number of direct pathways through which exposure to chronic or toxic stress can get into the body through activation of stress biological pathways. So quick biology lesson, what does this look like? So there are two major stress axes within the body. The first one is part of our nervous system, specifically the autonomic nervous system. And more specifically in there, the autonomic nervous system consists of the parasympathetic and the sympathetic nervous system. And the sympathetic nervous system is involved in our stress response. So what happens is that you perceive something stressful, these nerves, the sympathetic nervous system nerves are activated, and they start spitting out epinephrine into the tissue. They also innervate your adrenal gland, which sits on top of your kidneys in the back. And that causes your adrenal glands to start spitting out epinephrine into your circulation system. And that goes out and causes a stress response. The other axis that we talk about is this hypothalamic pituitary adrenal, or the HPA axis. So this one, something stressful happens, your hypothalamus is activated, it starts spitting out a hormone called CRH, that acts on your pituitary gland. Your pituitary gland starts spitting out another hormone called ACTH that floats around in your system until it reaches again your adrenal glands on the back of your kidneys, and that causes your body to start spitting out another stress hormone called cortisol. So in a nutshell, something stressful happens, you perceive it, you're in danger. There is an immediate response from your sympathetic nervous system that starts pumping out epinephrine. This is responsible for those jitters that you normally get, the flop, sweat, the butterflies in your stomach. And then just because it takes a little bit longer for these hormones to get around the body, cortisol starts coming online a couple of minutes later. And collectively, these two systems result in and support what's called the fight or flight response. And this is a multi-organ, multi-physiological response, the purpose of which is to mobilize whatever energy or resources you need to get yourself out of that dangerous situation, whether you have to run away or fight your way out. So it affects your heart, starts pumping blood. It affects your digestive tract, shunts blood away, gets your muscles primed to run, gets your immune system ready for injury. And I just want to say that these axes in themselves are not, this is not a health problematic process. It's actually quite adaptive. To get the resources that you need to get out of a dangerous situation is very beneficial. The problem more arises when you either get multiple hits of the stress response, or you are dealing with a chronic or toxic stressor that you can't escape. The system is meant for short bursts. And if the body is forced to continue this level of activation over long periods of time, it leads to these wear and tear adaptations that can lead to disease and other problems down the line, how the theory goes. Now, while stressor can activate these responses, things in your environment that can block or buffer that stress response can help keep this process from starting in the first place. So for example, in the context of the attach intervention, even though we're dealing with families that are living in potentially exposure to toxic or chronic stress, but a positive parenting relationship could potentially help buffer the experience of that toxic or chronic stress, which helps regulate these systems better. And you'll have noticed that there's a little cell down here, that the immune system is part of this, is one of the systems that's affected by the stress response. So why the immune system in particular? And it's not just because that's my area of study and I really like the immune system. The immune system is a very important physiological system. It's a little bit hard to pin down because unlike, lung tissue or liver tissue, you can't really point to one part of the body and say that's your immune system. There are parts of your body that are immune system organs. So for example, your tonsils, your spleen, and your bone marrow. But for the most part, your immune system consists of this collection of cells that just kind of float around and patrol your body. They're kind of spread out all over the place. And the immune system has two major purposes. So the first one is that of protection. The immune system is a very powerful system. We're talking about cells that can actually blow up other cells. They are able to recognize what should not be there or what's not you or what's damaged and go in and remove it. So whether that is bacteria, whether that is a cell that's infected by a virus, whether that's cancer, whether that's an organ transplant, these cells are able to say, this does not belong here, we're going to get rid of it. The other major purpose of the immune system is that of repair or healing. So if you are injured, even if you're not exposed to bacteria, if you are bruised, the immune system is what is going in there to repair that tissue damage, remove the damaged or dead tissue, and get you back to your pre-injured state. So two very important functions of the immune system. Now, another word that you commonly hear connected to the immune system is this inflammation or inflammatory response. So if the immune system is the thing or the object, then the inflammatory response or inflammation is the action. It's what the immune system does. Another way to think about this is that the inflammatory response is how the immune system talks. immune cells spit out a number of hormones, proteins called cytokines that allows it to talk to other immune cells and other parts of the body in order to, let everyone know what's going on in terms of the injury or the infection, and to orchestrate that overall immune response. And based on what I've told you, it's no surprise that the immune system is very tightly tied to disease. So pretty much any disease you can think of is going to involve the immune system and inflammation somewhere in that process. So from dementia, cancer, allergies and asthma, diabetes, heart disease, liver disease, systemic infections or sepsis, the immune system is going to be involved somewhere in here. So when it comes to understanding health or a mechanism towards health, the immune system is very important. And more than that, we also know that chronic stress is also related to immune activity and immune disease risk. So bringing it back all together quickly. So the central nervous system is part of what helps us talk to the physical and social environment, what's going on, what's out there. We know from the stress axes that the central nervous system can talk directly to the immune system through these sympathetic nervous system and the hypothalamic adrenal HPA axis. We also know that the immune system can talk back to our brain through these inflammatory responses and these proteins that it spits out. So what this all means is that if something scary or stressful happens within our environment, that is picked up by our central nervous system, which then communicates that to our immune system, and that causes a change in how our immune system behaves. So if we're talking about acute stressors, that gets the immune system primed and ready to go for whatever threat we're having to deal with. So you can see that if you're potentially going to be injured, having an immune system that's ready to jump in there and deal with whatever is coming your way could be beneficial. But in the context of these toxic or chronic stressors that you can't really escape, things start to get a bit problematic because the immune system is a very powerful system. It is potentially dangerous. It can, like I said, blow up other cells. So if it is activated for long periods of time, sometimes adaptations can occur that cause the immune system to behave in ways that are not adaptive, or potentially it causes it to be activated when it should not be activated. And that leads to a whole bunch of disease issues later down the road. That's how stress can get under the skin and start affecting your immune system in a way that relates to these diseases. So the immune system is important. We know that it can hear stress and it reacts accordingly. And we know that exposure to toxic or chronic stress can potentially affect your risk for inflammatory diseases later down the line. So it's important to understand and look at the immune system from a mechanistic perspective to understand what's going on and see what we can do to start interrupting these links between chronic stress and later risk for disease. So there's a number of different ways that we go about measuring the immune system and immune activity. So just a quick list going from very broad down to more specific. You can look at evidence of infection or inflammation-related diseases. That's very broad overview, but it's a little bit like closing the barn door after the horses have escaped. Someone is already ill, injury has already happened. But there are some preclinical things that we can kind of look at instead, where we're starting to tap into what's happening before disease happens and where we can intervene before damage happens. So for example, we can look at immune action. So for example, how quickly wounds heal is a good indicator of how our immune system is doing. We can look at immune cell activity in vitro. So for example, you might not be able to expose a human being to a certain disease or a certain pathogen, but you could take a sample of their blood, put it in a dish, expose that to whatever pathogen it is and see what their cells do. You can also do things like look at immune cell counts, how many of each kind of cell do we have. You can look at immune biomarkers or proteins that are floating around. Again, this is part of that inflammatory response, so you can get a sense of what the immune system is doing based on what signals are floating around in the body. And you can also take that one step further and look inside the cells at these intracellular activities to get a sense of how the immune system is activated and what it's doing. By example, looking at RNA and DNA. So I'm going to do a quick little talk on RNA because Sarah is going to be talking about the DNA portion of it. So what the heck is RNA? RNA stands for ribonucleic acid. And if you think that sounds an awful lot like DNA, then you would be correct. They're very closely related molecules. Specifically, RNA is involved in this process called gene transcription and translation. So in a nutshell, when you think about how a cell works or regulates itself, the DNA at the center of the cell is kind of like the cell's information library. It contains all of the information that a cell needs to do whatever it needs to do. But that information has to be translated into something that can be turned into an action in order for it to be useful. And it's this transcription and translation process that allows that to happen. So in your nucleus, your DNA is first copied or transcribed into an RNA. So it's transcription. So think about the scribes who are, medieval scribes that are copying out, versions of the Bible. The same sort of thing happens. You take the original information and you make a copy of that information so that you can manipulate it or use it to do something else. So that produces RNA, which is a copy of a particular gene that you are interested in, the cell is interested in working with. That RNA has been taken and it is translated from the language of DNA and RNA into the language of protein. And the protein is what then goes off and does stuff. It affects what kind of cell we're talking about, the activity that the cell is going to do. Pretty much any action a lot of the action is tied back to protein. But if we take a look at this full process then, if we zero in on the RNA, on these copies of information that are being produced from our library, from our DNA, this gives us a snapshot of what is going on in terms of that cell's activity. What is the information that's being mobilized? What is the cell's priority in terms of its action? It gives us a sense of what's going on inside the cell. So from a biomarker perspective, this is a very rich source of information that we can look at in terms of understanding what is going on in terms of cell or immune activity. The other reason that we like RNA, especially based on coming after Katie's talk, is that we can get at it using dried blood spots instead of a full out blood draw. Blood spots tend to be a much more community-friendly way to acquire blood samples because they are much less invasive. Instead of having a needle, you can poke someone a quick prick on the finger and collect drops of blood instead. And we can get measures of RNA more easily out of these than, for example, protein. Okay, so bringing that around, talked about the stress response, I've talked about the different ways of looking at the immune system, I've talked about RNA specifically. Let's talk about this specifically in the context of ATTACH. So this was a pilot project that we looked at for the last ATTACH pilot. My crazy idea. So we know that toxic stress exposure can affect child health across the lifespan. And we also know that parent-child relationship quality, which is what the ATTACH intervention is focused on, is also related to child health, specifically immune health in both of these situations. So as Nicole talked about last month, we know that the ATTACH intervention seems to be having a really good impact on child developmental outcomes. We then wanted to know, can we also extend that to a child's health outcomes? Is this also beneficial here as well? Yes, so does it have a positive impact on physiological outcomes as well? So as a way to tiptoe into this question, we partnered with Discovery House and using funds from the Alberta Children's Hospital, there were 20 mother-child dyads who were enrolled in the last of the ATTACH pilot interventions. And we took dried blood spot samples from them. Mothers and children were enrolled in either the intervention group or the waitlist control group. And we took blood samples after they had what the intervention group had completed their first intervention, but before the waitlist control started theirs. And we wanted to see whether or not there was a difference in their gene expression or RNA patterns based on whether they had completed the intervention or not. So we took the dried blood spots and we looked at RNA specifically. And more specific than that, we looked at something called the conserved transcriptional response to adversity. It is a terrible, I shouldn't say terrible, it's a very long mouthful name. But this is work that has been done by Steve Cole at the University of California, Los Angeles. And his work shows that when you are exposed to stress, toxic stress, chronic stress, your immune cell gene expression patterns tend to shift in a very systematic way, which is this conserved response to adversity. And specifically what this pattern looks like is you see an upregulation of pro-inflammatory gene expression. So these are the genes that are involved in getting your immune system revved up and ready to go and can be potentially damaging if they are revved up long term without something to aim at. And it's also related to a decrease in antiviral gene expression. So these are the genes that help you respond to things like the common cold or things like that. And collectively, there is evidence that this pattern is not a good sign. So for example, higher patterns of the CTRA have been associated with greater risk for viral infection, risk for chronic diseases like heart disease, diabetes, and things like shorter pregnancy. So this does seem to be a pattern that is part of that mechanistic chain leading from exposure to stress, changes to the immune system, and then potentially risk for future problems down the line. So just a quick look at the pilot data. We have the CTRA index here on the y-axis. Higher values are worse, and the control and the intervention group here on the x-axis. And what we found was that after being in the attach intervention, even for as short as 10 weeks, we were seeing a pretty big difference between the control group and the intervention group in terms of the CTRA. So the mothers and the children who had finished the attach intervention actually had healthier gene expression profiles in their immune system. They had much lower CTRA indices. So this is fantastic. This is preliminary data suggesting that attach can potentially also be protecting mothers and child's physical health. We're going to be building on this with the attach intervention that we're building on today. So anyway, just as a quick summary then, psychosocial experiences can get under the skin to affect immune activity and disease risk specifically. There's a number of different ways to look at the immune system, but RNA and gene expression is one way that we're doing that with this study through collection of dried blood spots. Specifically, we were looking at this pattern related to adversity called the CTRA, and we do have preliminary evidence that we're going to build on that attach could be related to immune cell gene expression and could be protecting health of moms and kids. So I'm going to jump over to Sarah. If you have any questions, please put them in the comment box at the bottom.
Thank you, Kara. That was fantastic. Yes, so Sarah, if you'd like to start.
Yeah, I very much would. Thank you very much. Yes, so that was a great introduction for me because I am going to talk a little bit about the DNA portion of why actually getting blood or blood spots from children can be so incredibly informational. When we talk about genetics, a great way to think of it as your DNA is the recipe for life, which makes epigenetics all of the notes and post-its and things in the margins that helps that recipe be specific and adaptable. And so we start life with the DNA sequence that we will have forever. And that doesn't change throughout our lifespan. However, epigenetics, or the alterations that can be made to your DNA that don't actually change the DNA sequence itself, work with that sequence to give a window of potential phenotypes and expressions. And these are persistent and mitotically heritable. And what that means is that one cell can actually pass those alterations onto a later cell, which means that even though something may happen in, for example, childhood, the epigenetic alterations that we see there could potentially last for your entire life. Now, today, and with the ATTACH program, we're really talking about social epigenetics, which is how life experiences, such as our social environment, our relationships with our parents, chronic stress, and things of that nature, are actually affecting the way that our genes are expressed through these tags on our DNA. And so our experiences affect our biology, and I think Kara did a great job explaining how that can happen. But then those biological pathways affect our DNA and subsequently our gene expression. And essentially, especially in childhood, but throughout our lives, our experiences allow us to learn from them. And those learning and memory ultimately result in behaviors and how we interact with our world, which then again affect the experiences we have. And this is ultimately the cycle of our adaptation to environment. And in the process of learning and experiencing the world, we can also have changes to our epigenome that essentially become our biological memory. Because these can be passed from one cell to the next cell, it's not necessarily just an experience in that moment. It is your biology learning from your environment. So though there are many forms of epigenetics, today we're going to be talking specifically about DNA methylation. So although your DNA doesn't change, there can be tags such as these methyl groups that are put on your DNA by machinery already in your body that essentially act like a dimmer switch, for example, on a particular gene. And the more methylation, the dimmer the expression, which essentially in that process that Kharah was talking about of transcribing, the more methyl groups, for example, the less transcription that you might have for any particular gene. One really great example of this is the agouti mouse. So each of these mice in the center of the screen are genetically identical. Really, the only difference that happened in these mice was the diet of their mothers, and whether or not their mothers were fed really methyl-rich food, which ultimately resulted in the difference that you see on the right of whether there was methylation in the promoter region of this agouti gene or not. And what you can see is that if there's a lot of methylation, then you wouldn't necessarily get a lot of transcription. So the higher the transcription of the gene, the different the phenotype. And the less the transcription of the gene, you actually are seeing a completely different mouse, even though they are identical in terms of their DNA. So these two mice, absolutely identical. One is orange, obese, and prone to cancer. And the only difference is the methyl groups that are on the promoter region of one gene. So clearly there is an effect of these DNA methylation tags. One area that we know, one environment, in addition to diet, that we know has an effect, are these early caregiving environments. And in fact, in the history of DNA methylation research, this is something that was one of the first questions really asked and has been asked throughout the course of our knowledge of social epigenetics. And one of the first studies to really look at this that I think is very illustrative is actually in mice, where one set of mother and child were compared to another set of a mother and child. And essentially the thing that they differed on was the caregiving sensitivity and attention that the mother gave the child. In this particular example, because they are in rodents, that would be licking and grooming of the child. And so what they found is that mice that were particularly attentive to their children, the offspring of those mice had differences in the DNA methylation at stress genes. In this particular case, in the glucocorticoid receptor, which is a gene involved in that stress response that Kharah was talking about earlier, and which ultimately ended up in different behavioral phenotypes of when those offspring grew up. They themselves either were more or less attentive to their own offspring. And this process of adaptation to the environment, social or otherwise, comes as a combination of the DNA, the underlying DNA that doesn't change, that we are born with and keep for the rest of our lives, along with the experiences that we have. And it is important to note that childhood in particular is sensitive to different experiences and exposures in terms of what our DNA methylation actually put onto our DNA and then kept throughout our lives might be affected. It is far more variable in the early years of life than it is later on. In addition to methylation in and of itself, and looking at a specific site where DNA methylation may be placed or not as that kind of potentially dimmer switch, we can also use a fantastic aspect of DNA methylation to look at immune cells in children as well. So because DNA methylation is essentially saying, here is what I want you to actually be transcribing from your DNA library, it is very specific for whichever tissue you're looking at. It is the thing that makes a skin cell, or a brain cell, or a blood cell. You know, every single cell in your body has your entire DNA sequence, and yet obviously they're very different. And so it's through epigenetics that process happens. So as an individual, you may have some DNA methylation that is the same across multiple tissues, but you will also have some that's very different depending on the tissue that you're talking about. We can use that to actually look at something like blood, which has many different cell types in it, many very specifically related to the immune system, which is something that we're very interested in, and use that aspect to say that these different immune cells have characteristic patterns of DNA methylation that makes them that particular type of cell. And so when we have DNA methylation from different people, we can actually look at the different proportions of immune cell types in one person to another and compare how their environment might be affecting the proliferation of a particular type of immune cell. The other thing about DNA methylation that is very well studied and well established is its relationship with aging, which gives us yet another opportunity to examine how these environments and social and chronic stress and things like that could be affecting our biology and that biological memory to get under the skin. So while from individual to individual, there will be a lot of differences in DNA methylation as well as the underlying DNA itself. There's also, we have found a really well-established pattern of there are certain sites in your DNA that change very specifically with age. And we can predict that because regardless of which individual you are, with time, things are changing in a predictable fashion. So we can take those sites and put them together to make essentially a biological age. And we can calculate what your age is according to the DNA methylation that we see on your DNA. Now, if your biological age and your chronological age are the same, then you're aging biologically at exactly the same rate as you are aging chronologically, which is great. But if you're actually biologically older, that is a sign of you being exposed to certain risk factors like chronic stress. However, if you are biologically younger, that can also be associated with protective factors. For example, interventions, social interventions, and interventions that prevent stressful environments from maybe taking hold. And there are well-established long-term effects. It's this concept of the social determinants of health and disease that can be very well illustrated in biological aging, in that people who age biologically faster have differences in aging and longevity and disease and health outcomes throughout life, but especially in older age. And we do see that people who are aging more biologically quickly are at a disadvantage, especially in older individuals. So in conclusion, thinking about the ATTACH program and how we want to use these blood samples and this DNA methylation information, firstly, we can look at the specific changes in DNA methylation. Where are they? What genes are they associated with? And how do they relate to the transcriptional changes that we see? Specifically to look at the possibility of long-term changes and how we can affect those that may last throughout people's lives. Secondly, we can look at those immune cell proportions and see how they might be affected. And we can compare the function of those different types of immune cells that we can look at based on those markers. And then thirdly, we can look at potential changes in biological age and how that might lead to protective effects associated with an intervention like ATTACH. So thank you very much. And open to questions.
Excellent. Well, really good. Well, thank you very much to all of our excellent speakers. I did see we had one question for Kara from Susanna, and she asked, my question is, how many times did you take the dry spots? Just once per group or before and after?
So this was, and I think you participated in this, Susanna, which is a good question. We ended up just, for the pilot project, we only took samples once. So sorry, it's been a while. We took samples from the control group and the intervention group once. And then for the control group that went on to do the intervention themselves, we took samples afterwards as well. We did analyze both of those, but we ran into a power problem. So we were able to pick up on the difference between the two groups, because 20 individuals is unfortunately fairly small for this kind of study. But we weren't able to pick up, do any of the analyses we needed to do on the pre and post ones, just because, again, we only had, I think it was 10 moms and kids for that. So we weren't able to do that. Yeah, so we did take them pre and post for one group. For the current ATTACH project, we are going to be planning to collect samples before and after the intervention. We have more families enrolled in this, so we would have the power to be able to do those analyses that we weren't able to do with the pilot. Yeah.
Okay, excellent. Thank you, Kara. So we have another question from Nicole. So this is for Sarah. So great to see you. What do you think of the potential for social interventions to impact epigenetic aging or some other epigenetic outcome?
Oh, I think they're very good, actually. I can think of one study in particular that was looking at minority stress in adolescents, and what they found was that with greater social support, which was unrelated necessarily specifically to the variable of interest, that they saw better biological aging, which in this case was slower biological aging. They didn't see that increase that's normally associated with things like chronic stress. So in terms of, do we know that social interventions can definitely affect a biological age? Yes. I think a big question in the field right now is how much, if we are affecting children's biological age, will that ultimately snowball or not as they get older? Because the field just hasn't really existed. These clocks haven't existed long enough to know for sure what the ultimate effect of these interventions might be as people get older. However, there is good evidence that these differences could be maintained over time. And so something like a social intervention, I do think that there is, especially in children, a really good chance of finding something pretty significant. Good question.
Excellent. Thank you, Sarah. Well, I don't know if I'm speaking for everyone, but we're certainly all interested in the aging process. So all this evidence really helps all of us. So we are at 1 o'clock, but we have one more question, again from Susanna, and this is for Sarah. Could you please explain exactly what is DNA methylation and how often does it happen in a cell?
Really good question. So DNA methylation is the deposition of a methyl group, so CH3, onto a base pair in the DNA sequence. So there are 4 base pairs in DNA. So specifically the cytosines, usually when followed by a guanosine. So CPG is often how it's referred to. So there are actually transcripts and genes in your DNA that are specifically for putting methylation onto a cell. And a lot of this happens very early in development in terms of the actual, knowing that an eye is an eye and your brain is your brain. But it could actually happen throughout your life, and you can put on new DNA methylation or remove DNA methylation throughout your life. And there are processes for both. There's a great deal of DNA methylation in any given cell because there is so much DNA in every cell that the methylation that's used to either upregulate or downregulate a particular gene, there's a lot of it. But there are also other epigenetic mechanisms, for example, how histones, which are the sort of proteins that DNA is wrapped around, how tightly or not that they are wrapped, and particular chemical groups that can be added to that. There are several different types of epigenetic mechanisms, and DNA methylation is just one. But yeah, in general, I would say there's quite a bit of DNA methylation in a cell. And for our purposes, the way that we measure DNA methylation at this epigenome-wide level is using microarrays, which means that we measure DNA methylation at approximately 865,000 sites in your DNA. But of course, there's much more methylation in a particular cell than that. Hopefully that's helpful.
So when I think of DNA methylation, and maybe I've got it totally wrong, but I always think of like a coating on the DNA that, like, if there's more methylation, then it kind of helps the connectivity better, and if there's less, then like it slows things down. But I don't know, like that's just,
I mean, it does actually work through charge, right? Like it because our DNA methylation, or sorry, our DNA has a charge to it, right? Like it's negatively charged. And so in the same way that magnets work, these chemical groups, or how tightly it's packaged, is actually dependent on how strong your polarity is. So there are other things that can be added, like acetyl groups that have an opposite charge that make it kind of unwind a little bit and make it easier to get to. And that's essentially how the dimmer switch works, is it makes it either easier or harder to get to the DNA that you need.
excellent. Well, we are almost at 1:05, so we've kept everybody a little bit longer, but I just want to thank everyone for joining us for this fabulous webinar, and really thank you so much to our 3 wonderful presenters. It was so interesting. Each of your talks was so individually interesting, and yet they all kind of, you know, coupled together. So that was really nice. So thank you all, and we hope that you'll be back. Did you want to say something quickly, Kara, about the May webinar, just for everybody that's still on?
Yeah, so I'll send an email out to the team, but just so that everyone knows, we had a bit of a schedule rearrangement. So in May, we're not going to have an actual webinar like this. It's going to be individual training from CIHR around sex and gender analyses in research. So we'll be sending information about that. But in June, we will be back to our regularly scheduled webinars, and we'll have Carrie Collier talking to us about the healthcare system and health policy. So stay tuned for that.
Excellent. Great. Well, thank you again, everyone, and everyone have a wonderful weekend, and we'll see you next month. See you later, everyone.